The Great Journey of Meiosis. What is meiosis? Meiosis is a specialized type of cell division that produces gametes, which are sex cells. In meiosis, the product is 4 haploid cells, which is really just sex cells that will create a new baby. Meiosis is important because it reduces the chromosome number in half, meaning that when 2 gametes meet, one comes from your dad and the other one comes from your mom. Together they will create the full number of chromosomes needed to create human life. Meiosis, the process. The beginning stage of meiosis is prophase 1, in which there is a nuclear membrane and spindle microfibers in the centriole. The cell has 2 N chromosomes, which means there's a double chromatid, and N is the haploid number of chromosomes. Homologous chromosomes pair, which is called synopsis, and homologous chromosomes are similar in shape, size, and structure, and have the same genes at the same locations, but not the same alleles. Crossing over then occurs. The second in the process of meiosis is metaphase 1. Metaphase 1 is when the spindle microfibrils move homologous pairs to the equator of the cells, and in the red and blue you can see the bivalents aligned on the equator. The orientation of paternal and maternal chromosomes on either side of the equator is random and independent of the other homologous pairs. The third part of meiosis is anaphase 1. So there you can see the homologous chromosomes being pulled to opposite poles. Homologous pairs are separated, and then one chromosome of each pair is moved to either pole. The fourth is called telophase 1. This is when the cell has divided across the equator. The chromosomes then uncoil. During the interphase that follows, no replication occurs. Reduction of chromosome number form diploid to haploid, and that is complete. Cytokinesis then occurs. Then this process occurs again. So prophase 2 is when the chromosomes still consist of 2 chromatids and condense and become visible. The second part of this process is then metaphase 2, where the bivalents are aligned at the equator. Anaphase 2 is when the centromeres separate and their chromatids are moved to opposite poles. Then we go into the very last stage, which is telophase 2. The product of telophase 2 is 4 diploid cells. We have the chromatids at opposite poles, the nuclear envelope is formed, and cytokinesis then occurs. Another thing that might happen is random orientation, which is when genes from each parent cross over to each chromatid, and that promotes genetic variation within the zygote. Thanks for watching!